Hello students, in this video let us discuss some important points about the amino acids. So amino acids are responsible for the formation of the proteins. Okay, generally we are saying that proteins are constructed by the amino acids. Okay, there are different type of amino acids are there, alpha amino acids, beta amino acids, okay, gamma amino acids, but all proteins are constructed by the alpha amino acids. Okay. So here, among the number of amino acids, generally we are studying 21 amino acids and their structures in our chapter, that is proteins and that means amino acids and protein chapter. We are studying the 21 amino acids. Okay. So among the 21 amino acids, so generally all amino acids are optically active. The glycine is optically inactive. Okay, all amino acids are optically active except the glycine, because the glycine has no chiral carbon, so it is an optically inactive amino acid. Okay, that is the glycine. Next, remaining all are optically active. Okay, remaining all amino acids have at least one chiral carbon. At least one chiral carbon. That's those are all the optically active. But two amino acids having two chiral carbons. Okay, two amino acids having two chiral carbons. For suppose this alanine has one chiral carbon, this cysteine has a one chiral carbon, this proline has a one chiral carbon. But two amino acids having two chiral carbons, that is one is threonine. Okay, threonine has a two chiral carbons. So this is the one chiral carbon, this one also another chiral carbon. This one has a two chiral carbons. Okay. Next here, isoleucine. Here this is the one chiral carbon, this is the one chiral carbon. This is the hydrogen, methyl, ethyl, and this group totally different. Okay. So threonine, isoleucine have two chiral carbons. This threonine and isoleucine have two chiral carbons. Remaining all amino acids, okay, 21 amino acids, each one has a one chiral carbon except the glycine. Okay, glycine has no chiral carbon. Okay, that is optically inactive. And next, one more important point is, all amino acids have capital L configuration. Okay, all amino acids have capital L configuration. We know that relative configuration, okay, DL notation. The DL notation is given for the bottommost chiral carbon. Of course, here in this case, if a hydrogen on the right side and here NH2 on the left side, so it is L amino acid. All amino acids NH2 on the left side in a 2D Fischer projection formula. So all amino acids have the L configuration. All amino acids have the L configuration. Okay. And next, one more important point. In all amino acids, this chiral carbon configuration is S actually. Okay. That chiral carbon configuration is, let us check this one. So NH2 first priority, here carbon, but carbon furtherly attached to the oxygen, so second priority, this one is the third priority. So 1, 2, 3 clockwise direction, but least priority group on the horizontal line. So its configuration is S configuration. It is absolute configuration. Okay, that is the absolute configuration. So all amino acids have the S configuration, but one amino acid, that is cysteine, has the R configuration. Why it has a R configuration different from the remaining all amino acids? Because of the SH group. Okay, the sulfur has a higher atomic number. Here NH2 first priority. Here carbon same. So proceed with further atom. Here oxygen. Here sulfur. Okay, sulfur atomic number higher than the oxygen. That's why this one getting a second priority. Okay, that is getting a second priority, and this one is the third priority. So here 1, 2, 3 anti-clockwise yes, but here least priority group on the horizontal line. So R configuration. Okay, cysteine has a R configuration. So the important points are the all amino acids have L configuration, relative configuration, but all amino acids absolute configuration yes, but one amino acid having a configuration R because of the SH group, that priority is changed. Okay, direction is changed. It is a anti-clockwise direction, but hydrogen on the horizontal line, so take the reverse. Okay, as per the rules we will discuss in a stereo chemistry. Okay, that's it. And one more important point is The amino acid which has a thiol functional group, the amino acid which has a thiol functional group is cysteine. Okay, that is also one of the previous JEE mains question. Okay, at the time of offline session, it is a mains question. Okay, which amino acid has a thiol functional group? That is a cysteine. Okay, that is a cysteine. Okay, and next, all amino acids are the primary amino acids. All amino acids are the primary amino acids. That means here all amino acids having the NH2 functional group. Okay, all amino acids having NH2 functional group, but one amino acid having a NH functional group. That means here it is a secondary amino acid. This is secondary amino acid. Simply it is called as a amino acid. This is simply called as a amino acid. Okay. So because of the NH functional group, it is a secondary amino acid and also called as the amino acid, and these are the amino acids. Okay. Yeah. Here in this case, one more amino acid which contains sulfur is the methionine. But here in this case, this is formed in a as a ether functional group. It is a thioether functional group. But here this is a cysteine. This cysteine SH functional group, that is nothing but thiol functional group. Generally here, the protein which contains cysteine amino acid after intake, okay, after intake into the body, it is dimerized. Okay, cysteine is dimerized. Its name is cysteine actually. Its name is cysteine. So here cysteine has SH functional group. Cysteine has a here that means it is a peroxide, thioperoxide group. Okay, SH linkage is present in a cysteine. Here cysteine contains SH and methionine which contains the thioether function group. Okay, that's it. So in this case, even sulfur atom is present, its configuration is yes, it's not changed because first priority, here in this case second priority, because this carbon is immediately attached to the oxygen, but this carbon immediately attached to the carbon. So based on the oxygen carbon priority, this one getting a second priority, this one third priority. So 1, 2, 3 clockwise, but take the reverse for S configuration. Okay, even sulfur atom is present, its configuration is a methionine, S configuration. Okay, but cysteine has a R configuration. That's it. Okay, so the difference between the cysteine and cysteine actually. Cysteine is a dimer of the cysteine. And next, it is a tryptophan amino acid. The amino acid which contains indole ring, is a tryptophan. The amino acid which contains indole ring is a tryptophan. Yeah. Next here, we have three amino acids which are basic in nature. There is the arginine, imidazole, and lysine. These are the basic amino acids because more number of NH2 groups. Okay. So in this case, this is the more second basic site. It is the first basic site. Okay. These are the two basic sites. Yeah, two are the basic sites. Okay. So in this case, arginine amino acid. This group is guanine group. Guanine group. The amino acid which contains guanine functional group, okay, is called as a arginine. Okay, guanine group containing amino acid is the arginine. Next here, this is a imidazole amino acid. Okay, sorry, it is a histidine amino acid. Okay, this is the amino acid histidine. But here, the amino acid which contains imidazole ring, this is the imidazole ring. The amino acid which containing imidazole ring is a histidine. Okay, so here in histidine, it is a basic site. It is a basic site. Between these two, it is a more basic site because this is a sp2 nitrogen. sp2 nitrogen. It is a sp3 nitrogen. sp2 nitrogen somewhat more electronegative than the sp3 nitrogen. That's why here in this case, more electronegative nitrogen atom holding electrons. That's why it is a less basic than this one. And now here, this is the nitrogen, this is the nitrogen, this is the nitrogen. Among these three, this nitrogen least basic because this lone pair of electrons are involving a resonance actually. Okay, that's it. So first basic, second basic and least basic nitrogen in a histidine amino acid, and this is the imidazole ring. Next coming to the lysine. Okay. So in this case, of course, it is also basic amino acid, no specialty, but this one histidine ring, this is a guanine function. Okay, that's it. And next here one more point. This is the basic nitrogen and this is the basic nitrogen. Okay, but here in this case, these lone pair of electrons are delocalized with this pi bond. Here lone pair of electrons are delocalized with this pi bond. As a result of the plus and effect of these two nitrogens, electron density on this nitrogen increases very much. That means it is a electron rich nitrogen, so it is the most basic than this one also. Okay, this is the most basic because of the more electron density. That electron density is increased by two strong plus M groups. That's why this one has a more electron density when compared with this one. It is the most basic site in arginine amino acid. Okay, that's it. Okay, so next here for remembering essential amino acids. Generally, amino acids are the two types: essential amino acids and non-essential amino acids. Okay, so for essential amino acids, dietary intake is necessary. Okay, so for remembering all essential amino acids, it is one of the small technique. So here that is a MTV high tech pizza. Okay, so here in this case, M means methionine. This is a methionine. T means threonine. T means threonine. V means valine. HI means histidine. Okay, HI means histidine. Okay, next here TEC means tryptophan. This is the tryptophan. Here P means that is a phenylalanine. This is a phenylalanine. I means isoleucine. L means lysine. Here L means leucine actually. It is a arginine actually. This is a arginine. This is the leucine. This is the lysine actually. Okay, lysine and leucine. I means here isoleucine. Okay, P means here phenylalanine, tryptophan, histidine, valine, threonine, methionine, and A means arginine, not alanine. So these are the essential amino acids. Okay, thank you for watching.